Now we're going to move on to the exhibition portion of my project. This is an example of stem cell being used as a shell for iPS differentiation, with the first topic being covered is safe and accurate drug testing. Research methods greatly utilize test animals such as rabbits when testing various drugs. Knockout mice are commonly used by mainly researchers as they are given a human disease for testing. These charts show the use of animals across medical testing, with 22% of testing being used in research and 10% being used for production of medicine. This is the overview of drug development process. This flowchart shows how a patient's cell can be reprogrammed into an iPS cell that can be used for drug development and genetic screening. Rats are given liver diseases that can then be treated with stem cells, very similarly to how humans will be treated in the near future. Instead of animals, researchers are beginning to look into regrowing human livers, which are the perfect environment to test and sample drugs, with the next picture being samples of liver buds growing in a petri dish. This is a proposed model of the liver generated by stem cells that can be used outside the body for testing or directly in a medical transplant. And the second topic of discussion is the treatment of diseases. Type 1 diabetes is caused by the inefficient creation of insulin, and type 2 is caused by a blocked insulin receptor. With this flowchart shows how researchers can introduce diabetes into a knockout mouse and can use the mouse's stem cells to differentiate pancreatic cells to now secrete insulin. And these graphs show the results of diabetic therapy through stem cells, as after 1 year of therapy, 25% of patients showed complete insulin independence after one full treatment. The next disease discussed is Parkinson's disease, where neurons and their receptors are no longer producing and receiving dopamine, a transmitter, through each other. With this sequence proposing how dead nerve cells can be implanted with stem cells, differentiated into new cells that will fully function and now transmit new nerve signals, with the next picture showing how a neuron can be fixed and connected once again. The left pictures show normal mice with complete function and activity, and the right side shows how knockout mice with Parkinson's have little to no activity. This is a recent study of Parkinson's stem cell patients where 36 of 75 patients showed great improvement after their treatment. These graphs show how stem cell therapy can drastically improve both serotonin and dopamine levels in Parkinson's patients. And overall, researchers are now able to take an inter-blastocyst cell, differentiate it into specific cells of the body, and can now replace inefficient cells that can produce and function as normal healthy cells. With the final disease discussed is heart disease, which is now a major basis for many research projects, as bone marrow stem cell differentiation is showing great improvements in patients. These graphs show how patients had a dramatic increase after their stem cell therapy in both heart function and blood volume levels. And finally, I will discuss how stem cells can help regrow organs and limbs. The main complex used to store stem cells at this point in the research is through a pig bladder, shown in the picture. This series of photographs are of a severed finger that was completely regrown, including now, through the use of a powder containing a mixture of stem cells and pig bladder. This amazing picture features the first toddler to receive a stem cell organ transplant, as the 2-year-old was born without a trachea, with the next picture being the trachea produced from stem cells. Researchers are now able to produce organs such as kidneys that are fully functional when given the correct biological sequences in labs. And this is the first kidney transplant with a stem cell produced kidney. The transplant was 100% successful and will not be rejected from the body since its original cells from the patient's body. Researchers have even produced functional ears along with other complete cartilage body parts. And other less important stem cell applications include regrowing hair on bald heads, where skin is removed from the bald area, stem cells are introduced into the cells, and then the hair follicles regrow. And the next picture shows the short-term results. Stem cells found in dental pulp are now being removed and saved by researchers to then be differentiated into other cells further in life to treat cancers and diseases, with the procedures having minimum pain and the stem cells showing high regeneration capacity. Picture a teeth being regrown from both dental pulp and even patients' urine, which also contains stem cells that researchers can differentiate. With this being the final product of teeth regrowth with the use of stem cells, providing a new avenue for the dental industry. Stem cells can also provide therapy for both patients by harvesting stem cells, then differentiating them into healthy cells, which are applied to the site for complete regrowth. This is a skin gun that is given a new opportunity to burn patients by basically being able to spray water and a stem cell solution onto injured sites. Patients are seeing great results with the stem cells showing complete regrowth even after the worst burns, with results coming almost overnight. This concludes the exhibition portion of my program. Thank you for your time and attention.